All right, I guess we can get started, no? So, welcome to everyone. It's a pleasure to open this Instruct workshop today. It's really meant for exchanges across all the CryoEM facilities and various centers that contribute either directly or join here to discuss. And the idea is really have an open spirit, have time for questions during the sessions and also in the breakout sessions, as you will see, so that we can see what is developing, where the field goes, where we can put efforts to improve things, either in terms of instrumentation, either in terms of image processing, for example. And it's really in that sense that we want to keep it open. And actually the sessions will also be broadcasted afterwards. All right, so there have been 2 workshops like this previously in the IBIC 2 years ago and last year in Nissen in the Netherlands. Instruct Centers, this is not just by chance, this is really an organization for the European community and beyond to be able to access high-end facilities. And it's not only in CryoEM, as you will see. I will give an overview of what is Instruct bringing to the community in terms of all the access possibilities and the various tools in there. So Instruct became an ERIC, a European Research Infrastructure Consortium, 2 years ago. And that makes a big difference because it's a European consortium that allows to organize research visits, training, internships, and R&D funding, for example. And it's across Europe with currently 13 member countries, among the first 6 that actually constituted that infrastructure is also the center here in Strasbourg. And from there it evolved now, so there are also associated centers for various fields. And you will see that in more detail. This covers all the aspects of structural biology and is really interfacing with other areas like imaging, like protein production facilities and so on. So the part of sample preparation is actually a key one because it is usually the bottleneck when you start a project and also when you run it at various levels, either in terms of protein production with various systems, including eukaryotic expression systems. Then crystallization is very often also something that is difficult, is a bottleneck by itself. So that's why the field is also moving a lot towards CryoEM. And nanobody discovery can actually help in that for crystallization, but also for imaging. We had a seminar from Anna Roussel, who is here today, yesterday. And so this is the typical part of sample preparation, which you can find across all the centers and it's available to the community. Then you have all the characterization and imaging associated, so analysis in the molecular sense, imaging with various microscope types, confocal microscopy, super-resolution microscopy, depending on the sites, mass spectrometry in some of the sites, molecular biophysics. So this is all the characterization methods which are available to the entire community. Yeah? And then the structural analysis, where you have electron microscopy, NMR, and Xray crystallography as the main driving forces. All right. So if you want to go there, and this is really an information also from Instruct itself, it's easy to apply. Through the ARIA web portal you can apply, and it's basically only whether, the question is whether it's technically feasible. No one is judging the science itself, but it will allow you to access and run your project in a given site. Okay, and so of course there's training associated, there are various workshops organized, like this one, as part of Instruct. There's internship possibilities, there's R&D funding, also there's calls for that, so just watch out. Next one is in 2020. All right, and if you want to look for more information, just look into the Instruct-ERIC website, where you can also register and apply and all that. Yeah? Good, and then you see also here is Instruct Ultra, so this is a specific European project associated to Instruct-ERIC, but the sense of that is actually to improve and facilitate user access. So that means either developing tools, for example, how to collect more easily data, wherever it is, I mean crystallography or EM, for example, or it is also to facilitate access for user communities which are not yet part So Instruct is actively exploring new sites in Asia, in the US, and so on. So in South America, typically, yeah. And this is the mission of Instruct Ultra, which is part of, it's a program, a financial program to run this as part of Instruct-ERIC. All right, I'm sorry. And so Instruct is hosted as an infrastructure here at the CBI, which we will visit later for those who would like to have a look. The site where we are right now is the IGBMC itself, and you have the mouse clinic here. So the CBI is placed in the center really also to centralize and to have interdisciplinary communication of various methods, and has its own research topic, as you can see here, around transcription, translation complexes, nucleoprotein complexes, RNA. And so you have Instruct that is hosted here, and as well Frisbee, which is the French Bruno, if you like, of the Instruct Infrastructure. And that's very important. I'll say a few words, because the mission is very similar. So it is really integration scale from the atomic level towards particle analysis, towards cellular analysis. And here you see already imaging coming in very quickly with phenotyping in here. And that is really to combine and go across. And it's not easy to do that, especially at every interface here. You need to do new developments. So some are being done in various centers, but the field itself really needs that. It's probably the most challenging part of it, is to do the interfacing right now. Okay, so in France, we set up that, it's 2009 we started that, and it started running really from 2012 on, the infrastructure for integrated structural biology with 5 centers. You have Paris, Strasbourg, Grenoble, Montpellier, and Marseille. Those 5 run projects for the French community. Among 2 are also Instruct Centers, so Strasbourg and Grenoble. And so this gives you a direct connection. That means any French lab can apply to go either to the French community, Frisbee, or via Instruct. Everyone should be aware of that. Vice versa, any lab that is member of Instruct, European-wide, can apply to any of the labs in France, provided they're Instruct centers. Yeah? So it means you get direct support, financial support to do that. And you see how important this link is between the 2 infrastructures from the applications and all the projects that are being run. So over the last period here, you see now reaching a plateau of about 800 user projects per year run. And training is actually very strong. And this is actually reaching sometimes the limits of a platform. Because when you welcome users, you don't only do the experiments, you are training the people, you are discussing the project, you are thinking of the science behind it to make it work. So that's very significant, in fact. Then there's lots of workshops that complement that, like the one here, but others as well, more specific, for example, for cell sectioning and other topics. And you see that countries here are submitting a lot of projects to Instruct, so it is actually providing a lot of help to the users. For Frisbee itself, you see that publications are running at a high level with a lot of highly visible papers. And what is important is that they're actually cited more and more. And this also shows that it has a significance in science to contribute, with a lot of international collaborations coming up and extending further right now. What we also set up as a French community is a national school that we organized around June once a year on the island of Oléron every year. And this is actually very dynamic and a unique opportunity to give teaching to master and PhD students mostly, or engineers who would like to get insights, get started, or get more into details of image processing or of NMR, of crystallography or biophysical characterization. That's basically the 4 areas that we cover there. Yeah? Good. And so now to come to the center here, there are 6 modules run throughout the platform. So you see it's not only CryoEM, even though we're doing a workshop on CryoEM, but it's very important, this link, this synergy between the various modules. So protein production facilities, biophysical characterization, crystallization facilities, Xray crystallography, including native phasing, NMR, CryoEM, single particle, but also tomography. Then super resolution setups with developments, I'll come to that later. And what is actually increasingly important is to have dedicated computing support, including on-the-fly processing. So with that, as a site here, we have over 200 access projects per year, with over 50 different research teams inside and outside combined. Okay. And so why is that running, and what is the concept behind it? Is really to create a synergy in the way we have conceived the building. And so we are on this side right now, but later when we will visit, you will see, when you enter on the left, on the ground floor, you have all these tools next to each other so that they can speak to each other. Yeah, so here's a Titan Cryos, there's a second room for Cryos, there's a Polara here, a FIP instrument, super resolution microscope, a D-Storm, G-STIM device, diffractometer, there's storage at liquid nitrogen of samples, there's computing resources, there's micromechanics to develop tools, there's an optics lab to develop new tools, and this is being done. I mean, there's a whole interface of it, yeah? And that's, I think, quite not only important, but also rather unique in the end to have that combination. Okay, and some scientific highlights from users and from teams here. That's a typical example of a combination of various tools. Crystallography here, this is run in the group of Marc Ruff in collaboration with the company, and Cryo-EM to visualize HIV complexes and how to inhibit them. So you have a whole combination here. Then there are some examples from my own team, what we have been working on human ribosome complexes with high resolution so that we could also resolve chemical modifications and inhibitors like here by Cryo-EM and do drug design based on that. Then there's a set of examples here from external users. Histone chaperone, this is with Grenoble, for example. This is another one from Grenoble. This is with Cambridge. And then you see numerous cases where actually the infrastructure is supporting new group leaders starting. And that's typical examples from Helgo Schmidt's group, from Albert Weichselbaumer's group, from Patrick Schulz, from Jean Caverly's group. So you see there's a lot of things happening with important contributions with various complexes. The latest one that just came out is from Valérie Lameau team on the gyrase complex. This came out where for the first time one can see the full complex of the gyrase and its DNA complex. Lately we also published something on CENT-A clusters. This is now with super resolution microscopy where you can actually distinguish these two. This is an epifluorescence and that's super resolution to analyze clusters of CENT-A. It's a histone variant in collaboration with Ali Amish at the institute here, and which allowed to actually reconstruct in 3D from super resolution data, provided you collect the data also with adaptive optics to get a 3D information and then segment the data. So that's typically something that is really increasingly important because we are going towards correlative imaging with that. And I think that is one of the points where the field can have a strong input in terms of developments. What we try to do here, for example, is developments around image processing, faceplate analysis, the super resolution microscopy applications, fibbing. One of the most tricky things is certainly still to produce lamella and transfer them and then do tomography. This is by far not yet a pipeline, and a couple of places are doing that. We will see during the workshop what is actually being developed right now, and it's very exciting to see. Okay, and so the spirit is really this combination, and it's much more than electron microscopy in an isolated manner. It is really integration, combination at the interfaces, and you see that especially fields like this, super resolution, are now coming into high resolution towards the 20 nanometer range. EM is crossing the whole range together with FIP analysis. Yeah. So it's a unique opportunity to go into this direction. Sorry. Okay. And for that reason, actually, in France, we set up a strategic roadmap to build up centers with high equipment. So that will be at CSRF at the Soleil synchrotron and here in Strasbourg for the National Center in Cryo-EM. The aim being to provide access, like we have started doing already, but extend that, on-the-fly processing, remote control, various tools like high-end cameras that are always evolving. That's another problem in our field. We will certainly discuss that during the workshop. Include FIB lamella and FIB SEM, high-resolution cryo-electron tomography, and all the aspects around it. Yeah. So the National Center will comprise high-end instrumentation and screening, electron microscopes, new freezing devices, super-resolution instrumentation and developments for corrective imaging, standardization of cryo-FIB lamella. This is really a challenge towards cryo-ET applications. Microelectron diffraction also is coming, and this we will implement in the future. Not only have storage and computing, but have really the dedicated developments associated to that. And it's on-the-fly processing is a key example, but it's more than that. It's interfacing with the tools I was mentioning. This will be hosted here at the Center for Integrative Biology at IGMBC and linked to the infrastructure, so that will be accessible. Frisbee as a French infrastructure, Instruct-ERIC at European level, Instruct Ultra as a project to promote access, and INEX Discovery, as we are now as a center becoming also an INEX center. This is with support from Europe, state and local government. Okay, and I think the field is really evolving a lot. I will finish this with these slides here. A few years ago, this was what you would get with Xray crystallography, and you can still do that, and it's very important to keep that tool. We should not just compare things, because for drug design, this is much easier to go for crystallization. But you see what the maps can look nowadays, and you can also do drug design provided the complex is stable enough to do that, and you push resolution. And this is a recent comment in Acta Chris. So you see this is a PDB contribution. When you look at large complexes, they are all here. So most of this is actually around 40 kD crystallography. And these ones are all cryo-EM based. And that's really clearly there's a shift, because now you see here the contribution of cryo-EM and crystallography to the PDB has just changed. Yeah? So this is 2016 about that. And you see how this now contributes to high resolution data as well. Okay, so with that, I'd like to start the workshop and like to give many thanks to Céline Klein, who's actually standing here at the door, and Marie-Christine Potesman, also sitting close there. They helped a lot to make this workshop happen, and I think we can just thank you in particular. Yeah. Absolutely. And then we have Alain, who is handling everything in the back, and he will be filming also and playing music and all these things, distributing beer maybe. Jean-Luc and Philippe. Then, of course, the platform. Many people from the platform are actually present, so it's a chance for you to have a chat with them. Catherine Birk is the coordinator of the platform. And the Department of Integrated Structural Biology, obviously. And the team members, many are sitting there, I see, who helped to set up various aspects during the meeting and to prepare things. So, with that, I'd like also to thank all the sponsors. It's very important to make it actually runnable, practically, to cover costs for the speakers, for the whole workshop and so on. So obviously the infrastructures, including Instruct Ultra, the companies, Thermo Fisher, Zeiss, JL, Quantifoil as well, and a company that actually emanates from our department here, Novellix, and the TEL, the CNS and INSERM, so that's medical research in France, and the University of Strasbourg. And INEX Discovery is coming very soon, so we look forward to that. And so the program is, after the welcome and just what I just did, is Luca Rima starting next to present the CryoWriter. So you will see developments on how to freeze samples. Then Rebecca Dilla from Nissan will speaker about the electron tomography workflows at Nissan. And then we have breakout sessions. And so you see, this is the first session today is sample preparation. Tomorrow there will be another 2. What we do after having these general presentations is breakout sessions. And we will split basically into 2 teams, working and then feedbacking, so we come back here afterwards, after some time. What we propose is that Rebecca is taking over the sample quality assessment later on, and Wim Hagen, you take the other group, so we'll discuss that later, just after the coffee break here. Yeah? And to see the overview for tomorrow, we have on-the-fly processing, data collection strategies, with Shausha from Cambridge, Pablo from Madrid, and Dimitri from Max Planck Göttingen. And then afterwards we have the breakout sessions, software and data management. And in the afternoon we have electron tomography and FIP with Philipp Erdmann here for in situ structural biology, hopefully towards high resolution tomography as we all hope. James from IWIC, also on FIP and tomography. Wim on data acquisition and various aspects of subthermal averaging. And Andreas Schertl finally will close the business with FIP analysis. And so with that, I think we can just start and with the next speaker. Maybe you have some questions of whatever type, scientific or organizational. Yeah. If there are no questions, then we just move to the first speaker then after this. I hope they are already loaded. Actually, if we could have also Rebecca's talk, I'm not sure that I've not seen her yet. Are you here? Ah, perfect, okay. Because it would be good to load both of your talks and we can just continue more smoothly afterwards, please. All right.